Okay, so do you three have any questions on what we talked about? Since Zach, I know I lost you about 3 minutes into the lecture last time.
I haven't had time to look at it again because I worked doubles all this weekend.
Okay. What about you?
No, I don't think I really had any. It was just my brain didn't want to work on.
I do not have any questions either, but if possible, could I leave a few minutes early today because they scheduled me at 3
Sure.
and I work in Bloomwood South.
Your
Traffic's
dad just
been
kinda
pretty,
hates you.
yeah. But they don't hate me, I just don't think they take the time to look at people's schedules. And
That's
You're
how
like,
my
oh, this
mom.
guy can work here. And you're
Yeah.
like, sure.
like, I have my unavailability set until 4 and they still scheduled me at 3, so
Sounds like most times.
yeah, can't complain though. Still a job.
I was asleep to the dairy this weekend. I went in Friday night and I worked Saturday morning, Saturday night, Sunday morning, Sunday night.
Just really bonded with cows.
I had one die on me too.
That's not fun.
I don't know why this
It was
isn't.
real sad.
Especially because dairy cows aren't supposed to die. So you get kind of attached.
Yeah. And then what really got me was when I saw her, like, she like picked her head up and moved one time real loud, and then her head just hit the ground.
What
and just
happened?
tears just started rolling in her eyes. I was like, dead. Like, I couldn't do anything at that point.
What was it?
I think she either had a heart attack or something internal messed up. I don't know. Because I went back 5 minutes after she died and her stomach was just as swollen as she could be.
Well, can't catch everything.
Some reason this doesn't want to. Connect. Of course, the time they decide to get all hip stops working. Okay. Let me think of some questions that I can quiz you all. Well, I'm getting this other one up. What's the common property all amino acids have?
OH.
So they have OH, what else do they have?
I think all I wrote down is they all have their own property. That's interesting.
What would they look like?
Was that Friday's class?
Yeah.
No, I wasn't here Friday.
They all have an amino group and yeah, the amino group, the COOH group, which I don't remember which one it is.
That's an acid.
And they're around the central carbon.
Right, what does an amino group look like?
H2N.
What is the thing that makes amino acids, that component that makes amino acids different from each other?
The R group.
R group.
So what group?
R group. When you get more than one amino acid, put together, what is it called?
Peptide? Peptide?
Peptide.
Peptide,
Yes. Peptide, tomato. When you make a peptide, what other thing that ends up being released?
Water?
Water. How many amino acids are there that make up protein?
20?
22?
22, I usually go by 20, but, how many are essential? 10. What's our acronym to remember?
Private Tim Hall? No,
Private Tim Hall.
That doesn't make sense.
So that is,
That's why you said military.
Right.
Okay.
so what does Private Tim Hall stand for?
Feeling, I don't know.
Phenylalanine,
Yeah, I just wrote the things to
valine,
threonine, trinine. Don't
so
remember.
threonine, tryptophan,
We can
isoleucine,
just memorize the 3-letter version, right?
yeah. Methionine, histidine, arginine, leucine, lysine.
Is it just PBT, Tim Hall?
What are the different structural levels of proteins?
Primary, secondary, tertiary, and quaternary.
What makes up a primary?
That's the peptide.
And our peptide is what?
The group of amino acids.
So a single chain, right? How long do those normally go? So if you end up getting more than 20, you move into our secondary. What makes up our secondary structure?
Is it 2 peptides?
It's hydrogen bonds. right?
You have hydrogen bonds, and they form 2 different things. We get 2 different types of secondary. If your group, in the Greek system, you may remember this.
Alpha and beta.
Beta, so you have alpha helices, so that funny A, alpha helix. And then you have beta pleated sheets. So these kind of go in circles like that, and then your beta pleated sheets go back and forth like that. What's our next level?
Tertiary.
Tertiary. What makes up our tertiary?
Disulfide bonds.
Disulfide bonds. So this provides our 3D structure. Now this you're not going to know, but I'm going to ask anyways. Do you know which 2 amino acids are going to be making these disulfide bonds? There are 2 amino acids that have sulfur. Okay, this one begins with an M, methionine. And the other one is cysteine. So those 2 make up your,
Cysteine is not one of the essential ones, right?
correct. Cysteine is what we call semi-essential, because methionine can make cysteine. So you can meet your cysteine requirement. So when you guys looked in your books for the amino acids, you sometimes may see TSAA. Any guesses what that stands for?
Total sulfur sulfide amino acids.
So this is because your methionine can make cysteine, we put them together and we call them our total sulfur amino acids. And the key is to at least have your methionine requirement met, because you don't want to put a bunch of cysteine in there to get your total sulfur amino acids up, because then you're going to end up with you're going to end up with a deficiency. But you can have your methionine meet your cysteine requirements.
So you don't actually have to add cysteine.
You don't have to actually add it. There are some protein products that have a lot of cysteine in it, but they end up being low quality protein, and that's because it's primarily all cysteine. One of those things is feather meal. So feather meal has a lot of cysteine. Your hair, your fingernails also has a lot of cysteine. So people that have curly hair, typically their hair protein has more cysteine in it, and that's what makes the curls, is those sulfide bonds as the protein keeps making.
So is that also like in, like antlers, horns type
Mhm.
thing? Okay.
Yeah. So nails, hair, feathers, hooves, horns are all going to be a lot of cysteine. What else did I wanna? So what makes up our quaternary?
That's a different types of proteins.
So we have more than one tertiary protein come together. What was the example that I gave you for that?
Hemoglobin?
Hemoglobin. So there are 4 proteins that make up hemoglobin. Why don't we want to feed excess protein?
It's a waste
More
of money.
money.
It's a waste of money. Why is it a waste of money?
Protein's expensive.
It's expensive.
They're not digested at all.
It's not that they're not digested,
They're not stored.
they're not stored.
They're stored, yeah.
So we can't store protein. So if you can't store it and you feed excess of it, what do you end up using it for?
Waste, fertilizer.
What's making the waste?
Feces.
The feces. So we're going to be breaking it down, right? We're going to take that extra amino acid, convert it to glucose. What do we have to take off to convert it to glucose?
Water? It's just-
No,
The acid,
not the acid part,
amino.
amino part. So that amino part gets excreted via what?
The pathway.
A pathway.
Is
So
it that
you're gonna be, for fish, what are you gonna make? What are you gonna do with that amino group?
one?
What do you make? What's the byproduct?
Oh, nitrogen or ammonia.
Ammonia. What do we make with birds?
Uric acid.
Uric acid and a little bit of urea. So that's all gonna be excreted. So anything extra, you're gonna end up breaking down using the carbon chains for glucose. The ammonia part is gonna be broken off and it's gonna be put in the waste. So then you get what potential pollution?
Nitrogen.
Nitrogen pollution. So that's why we don't want to feed extra of that. So then we have 2 different ways that we can calculate what we call our digestibility of amino acids.
Where you do a cystectomy or find the allele contents?
So that would be our ileal digestibility. And that is, so we're getting rid of all the, that's where we're getting rid of all the microbial changes, right? So microbes can take anything, a carbon and a nitrogen source, and they can make any amino acid and consequently any protein they need. So you can feed methionine. If it gets to the cecum, your microbes may change that to glycine. But if you look at just the feces, it's gonna look like you had lower digestibility of glycine, but higher digestibility of methionine, and that's not the case. So ileal digestibility is looking at what happens within the ileum before we get to the ceca, which is different than doing a total tract, which is collecting all the feces. So you can do it with cystectomy, where we take the ceca out. So why does that work in poultry?
They don't need the ceca to live.
They don't need the ceca to live, and what else?
I said it's like the pancreas and.
Not the pancreas, the appendix. And so, but there's also another key part that makes poultry different than let's say humans. So if I take my appendix out, I'm still gonna be able to have bacterial fermentation, right? Where is that gonna occur primarily in a person? Which portion of the GI tract?
The small intestine.
What portion does what happen?
Fermentation in a person.
Your large?
Your large intestine. What about poultry? Ceca. Do they have much fermentation occurring in their large intestine?
No,
Why?
it's only about 2 inches long.
It's only about 2 inches long. So there's not, and once it hits the large intestine, they don't have significant storage there. It passes through, their passage rate is actually really quick. From the time they begin to eat till the time it comes out, it's about 2.5 hours. So once it reaches the large intestine, there's not much time that is spent in there. So that's why we can do the cystectomy and take those out and get a good estimation of what the bird is actually using as opposed to what the microbes are doing. Does that make sense? Okay. Is this clarifying things for you, Zach? Tracking a little more? Okay. We have different types of end products when we break down our amino acids for energy. We have ones that are called glucogenic. What do you think glucogenic amino acids can result in making?
Glucose?
Glucose. So the majority of them are glucogenic, and then we have ketogenic amino acids. And our ketogenic amino acids make what? What do you think? So these make ketone bodies. So ketone bodies are the acetic acid, acetyl-CoA, and again, it's acetoacetic acid. So these things cannot go into the TCA cycle and make energy. So that's what ketogenic. So if you ever know anybody that's done a paleo diet or Atkins diet, a lot of times they run the risk of getting ketoacidosis. So what is that? They get a buildup of ketone bodies within their blood. And that's because they're using they have to break down the protein to make glucose, because those diets aren't, you have really low carbohydrate diets, right? That's the whole paleo and Atkins diet, and I think ketogenic. And so because you have to be breaking down the proteins, and you need those proteins to be utilized to be made into glucose for gluconeogenesis. When you're breaking down the proteins, some of the proteins going to have the glucogenic amino acids, some are the ketogenic amino acids. So if what you're eating has more of these ketogenic amino acids, you run the risk of getting ketoacidosis, which a lot of times when people first go on these diets, you'll notice that they aren't as quick-witted before, they can't think as clearly. Usually about a week into it, they just have a really hard time bringing up facts and remembering things. And that's because their brain is switching over to being slowly, their brain is actually starving, and the brain is slowly starting to convert to use ketone bodies. Yeah.
And what is a ketone body?
So this is the acetic acid and acetyl-CoA. So these are acetic acid makes acetyl-CoA. If you remember, acetyl-CoA is the main thing within the TCA cycle. So pyruvate's converted to acetyl-CoA, and then that goes to citric acid, and you start the cycle.
Okay.
The tough part is these molecules are so big that your gluconeogenesis is happening outside of the mitochondria, and these compounds can't get into the mitochondria. Where when you make these other components, these can transfer into the mitochondria and can make the TCA cycle. So you'll end up getting a buildup of these molecules. They're just really big.
Okay.
They're called ketone bodies.
And if they, let's say you have a balanced diet, but you're still getting too much ketogenic amino acids, can that be harmful, or is it only if you don't have enough glucogenic?
It's primarily if you don't have enough glucose. If you have enough glucose, you'll be able to maintain everything. And these will then go and help make your fat. When you break down fat too for energy, you also make these ketogenic or these ketone bodies.
Okay.
So that's the combination of whether you do the ketone diet, the Atkins diet, or the paleo diet. All of them are really low carbohydrate. It doesn't really matter if it's high fat or high protein. The consequences are pretty similar. The main ones that are strictly ketogenic are leucine and lysine. So you could see like tryptophan can be ketogenic. It can also be glucogenic. Your phenylalanine and your tyrosine are ketogenic or glucogenic. So leucine and lysine are the only ones that are strictly ketogenic. So this is essentially showing where these things can go in. The tough part is with these are broken down outside of the mitochondria. And so this molecule, acetyl-CoA, it cannot go into the mitochondria. The acetyl part's really small, it's just 2 carbons. But this coenzyme A is massive. Like an anchor, so that's what's controlling everything. Where all these other ones can go directly into, this is the TCA cycle, so it can have things moving around through here. So those are why those are glucogenic. So I mentioned this a little bit on Friday. So when we're formulating diets, we always want to do this ideal protein, or ideal amino acid concept. So what is that concept? That concept is where we are feeding each amino acid at the ideal amount that that animal needs at that particular phase in its life. So all of that ideal amino acid is set based off of lysine. So if we're trying to determine the requirement, so you can set ratios off in your formulation software and to say you wanted a percent of whatever your lysine is. So if your lysine ends up being 1% of the diet, so you're going to set all these other ones as a percentage of this lysine. So you can almost think of it as lysine is the currency for determining your amino acid value. So if you're going to think of it as a currency, you can think of it as the US dollar is the standard, right? Everybody around the world has an idea of how much the US dollar is, and then compared to the US dollar, how much is the yen, how much is a euro, and you can kind of think of it doing it that way. So lysine's 100%, so threonine is less than that. You can see methionine's even less. Our sulfur amino acids, which is our methionine plus cysteine, is more than methionine alone. Phenylalanine, or leucine's equivalent to lysine. So these are nursery pigs. So this is just the individual that came up with this theory is Dave Baker. He came from the University of Illinois. He is the protein amino acid guru for animals. He's the only scientist that's been inducted into the National Science Foundation that never got a federal grant. All his research was based off of corporate funding. But he got inducted into the National Science Foundation primarily using this concept. It's determining the amino acid ratios.
Is he still a professor?
See, he was on my master's thesis committee. He died 6 years ago of pancreatic cancer, maybe stomach cancer. But until the day he died, he was going through chemo. He was retired for 10 years before he had grad students. He was on my committee. He was one that never fully retired. I would call him institutionalized. So he was retired, but he had an active lab.
Gotcha.
To the very end. And you talk to anybody in this building and they all can contribute stuff that they know to Dave Baker. And he wasn't just a poultry, he was actually more of a pig guy, honestly. But he really did a lot of interesting research related to amino acids and proteins in livestock. So amino acids, as I mentioned before, we don't want to overfeed our amino acids because we're not going to improve our performance by overfeeding. Now, if you end up being deficient in something and then you get that amino acid up, you're going to see an increase in the performance. But overfeeding, if you're at the requirement, it's not the some is good, more is better. It's adequate is what we want, right at the requirement. If you overfeed, you're going to increase the nitrogen excretion, which is contributing to pollution, and protein is our second most expensive component in the diet, with energy being number one. So natural sources that we're going to put in our diets are anything that's a byproduct. So anything that has meal, soybean meal, that's a byproduct of the soybean oil industry. Distiller dry grains, byproduct of ethanol production. So poultry meal, meat and bone meal, trying to think of other, fish meal. So all of those byproducts. And this really is what makes animal agriculture sustainable. So many people don't like animal agriculture. They think it's unsustainable and we are misusing our resources, our limited resources here on Earth. But if you were to only have vegetables or crops to meet all your requirements, the majority of all the things that we are feeding, with the exception of corn, is a byproduct of the human industry. So if we didn't have animals to feed soybean meal to, what were we going to do with all the soybean meal? It would go into a landfill. So the cool thing about animal agriculture is it then makes something, a product, a byproduct of the human industry, to where we now feed it to this animal, and then we get the manure, and then what can we do with the manure?
Spray it.
Spray it on the field, which then allows the crops to grow. It re-nourishes the soil and provides nutrients for the crops. But you can't take soybean meal and spread that on the field and expect to get the same benefits. So that's in my opinion, animal agriculture really is making things more sustainable by my definition of sustainability. Our sources of amino acids, the quality varies. It really depends on the source. So if you think of back in the late 90s, early 2000s, when ethanol production really got going in the US. So we really saw Brazil. So Brazil has 85% of their vehicles are fueled by ethanol. And at that point in time, we really saw an increase in the cost of oil, petrol oil. And so people were putting pressure on the government, we need to find alternatives. And seeing Brazil had 85%, they were able to do this with ethanol. That's when we ended up, the federal government decided to subsidize a bunch of ethanol plants. So byproduct of ethanol plants is this distiller dry grains. And they needed to do something with these distiller dry grains. But the problem was, before this ethanol industry happened, before that the distiller dry grains was coming as a byproduct from the breweries. And there really aren't that many breweries compared to the byproduct of the distiller dry grains coming from breweries didn't even pale in comparison to what the ethanol plants were producing. So the problem was, we were having, depending on the time of year, when things were processed, which plant they came from, the quality of that product varied so greatly that nutritionists didn't want to take it. So it took probably about 5 to 8 years of doing various research studies and to where we got these ethanol plants to make a consistent byproduct. And it was similar with soybean meal. Nutritionists need a consistent byproduct so we can formulate our diets correctly. So we want to make sure that the quality is similar going from, because we want to be able to make one formulation and not have to keep reformulating every time we get a new lot of soybean meal or distiller dry grains come in. Because usually when that product comes in, it needs to be put in the feed mill right away so then we can make the diets. I can't necessarily wait to get the analysis back and make sure that it's meeting everything that I need. So we need the quality to be consistent. In addition, there are anti-nutritional factors that are associated with some of these. One of the components in soybean meal is trypsin inhibitor. So, and a lot of your legumes have trypsin inhibitor. Trypsin is one of those enzymes that your pancreas releases. So this is a pancreatic protease. So protease meaning that it breaks down protein, and it's coming from the pancreas. And this is the key thing that's starting protein digestion in the small intestine. So you have the pepsin coming from the stomach that's kind of breaking that quaternary and tertiary structure. We get long peptides. Our trypsin is making short peptides. So soybean meal has trypsin inhibitor. What do you think the trypsin inhibitor is doing? And why do you think soybeans evolved to have that?
Because they don't have a lot of protein.
Well, they evolved to have it because they have a lot of protein. So soybean meal.
Or the opposite, so they don't.
Yeah. So they're a good protein source. So you think of your beans, all your legumes. There's a lot of protein. If you're a vegan or vegetarian, you're going to eat a lot of soybeans. So they are a really good protein source, plant protein source. What do you think trypsin inhibitor does? We'll just flip these words around. What does trypsin inhibitor do? It inhibits trypsin. So if you're blocking the action of trypsin, what does that end up doing?
As it builds up protein since it's not digesting it.
So you can't digest your protein then. So this trypsin inhibitor is actually binding the trypsin, and you're not able to break it down because you can't get those single amino acids without this, what's technically the second step. Yeah.
Is there any, I'm sorry, I need to end the clutter word.
That's okay. We got it recorded.
All right, appreciate it.
So we have these anti-nutritional factors. The primary one that we're concerned about with soybean is trypsin inhibitor. So it's blocking trypsin, so we need to test for these, and maybe Friday we'll do it. I have some stuff that's called Soy Check that you can just check and see how much trypsin inhibitor that product has. So, yeah, Zach.
You said something about, was it Thursday at 12?
Yeah,
It's Thursday at 12. Okay.
Thursday at 12.
What building?
Scott, same place where we were before.
Okay, just the same place.
Yeah,
I wanted to make sure because I wanted to try and do it.
yeah.
You're
So-
right here, I think.
So if you go, you can either meet me at my lab, which is 247,
Okay.
or you can meet on the third floor by the bird wing. So we have other nutritional factors. Can you tell me another nutritional factor, Peter? Gossy Paul. And so then we also need to limit the fiber content, or know the fiber content. So what else do you know about beans? What's the fun little rhyme you learned as a kid related to beans?
Yes, with parts.
Yes. You wanna enlighten Zach?
Yeah, I never heard this
I
right.
can't remember it.
Beans, a magical fruit. The more you eat, the more you toot.
Okay, never heard that before.
So what causes beans to do that? It's the fiber that's associated with it. So all those things we have to be cognizant of using these natural sources of amino acids. Go until 2:45, right? Yes. Okay. However, because no protein source is ideal, there's always going to be, depending on the animal, some amino acids that are limiting. We also make these synthetic amino acids. So the most common ones we utilize, what are the most common ones that we use in our diets?
Lysine. Methionine, and then threonine.
So we added these. So by putting these in, we were able to, utilizing the synthetic amino acids, you can reduce your crude protein in the diet, but maintain the optimal growth of the bird. Because we're able to, you can almost think of it as a wine barrel, is the way I've had it described to me. So you have your wine barrel. So these are going to be so if this is my wine barrel, how full can I fill this without spilling over?
To this lowest one.
So we can fill it to here. So this would be what I call my limiting amino acid. But if our requirement is here, I have two options, right? I can increase the amount of soybean meal, which would bring this up to here, but then it's going to bring this equally up to there, right? So then we end up overfeeding, let's say that's glycine. So if I do that, that ends up contributing to what? If I overfeed all those other amino acids.
Obese.
Not obese,
Obese.
the waste.
Yeah, okay.
So that's contributing. We end up, if we have use our natural source of amino acids, so if it's normally here to have a lower crude protein, but I may need to put it up to here, everything's going to go up. So all of these other ones are going to be overfed. So what we do is we add in these synthetic amino acids. So then I'm able to, with the synthetic amino acid, just bring up these ones that are limiting. Does that make sense? So we're able to leave our crude protein lower, which is leaving our nitrogen level lower, and reduces our nitrogen pollution. Now, interestingly, real world situation is, so we can make synthetic lysine. That's pretty easy to make, and it can be in an organic manner. So you can get bacteria to make this. You can get bacteria to make this at a higher level. You cannot get them to make higher levels of methionine. So what that results in, methionine is truly synthetic. We are making it in a lab, putting these compounds together. Do you think organic people like that? Zach says no. Why? Can you give us a few minutes?
Because it's definitely not organic.
Because they view, since it wasn't grown from something, it can't be certified organic. So the problem is with poultry, especially laying hens that live a long time, you have the National Organic Standards Board is trying to limit this to 2 pounds for the life of a bird. So saying that you can only eat 2 pounds of synthetic methionine to be classified as an organic, if you're gonna raise organic eggs. That's not very much in the life of a laying hen. How long do laying hens live?
It's like 3 years.
2 years, like 105 weeks. So in a year, they end up meeting this. So, and you also need methionine to make, that's one of the primary amino acids in egg albumin. So if you want your nice, beautiful eggs with the thick albumin that's not runny, you need methionine, okay? So the organic industry is pushing that instead of doing what I have here in red, they want us to just feed more soybean meal to where we would have everything go up.
Then you're just overfeeding, right?
Means that the price of everything goes up.
So that's gonna increase our price of our eggs, because protein, again, is our second most expensive ingredient. And then we have, the other concern is now we've overfed all these other amino acids, and we make more ammonia within the
And then you have
house.
hot,
And you're not gonna be able to spread as much
Yeah.
of that manure on the field. So real life situation that Dr. Anderson and I are actually writing a grant for, trying to evaluate other potential natural sources that have high methionine. But the problem is methionine is so tightly regulated in your body, it's so critical, it's critical to make DNA and RNA. Would you have ever guessed that you need methionine for DNA and RNA?
Isn't it the stop amino acid? I just remember something else.
I'm not sure about that, but it's also, to make your purines and pyrimidines that are what your DNA and RNA is made up of, you need methionine. So it's your one carbon metabolism, methionine is critical for, and since that's so tightly regulated, no living organism can you trick it into making more of it. So that's the tricky part. So we'll end right there.